Gram staining. Gram staining is a method of staining used to distinguish and classify bacterial species into two large groups, gram-positive bacteria and gram-negative bacteria. The method is named after the Danish bacteriologist Hans Christian Gram, who developed the technique in 1884. Gram staining differentiates bacteria by the chemical and physical properties of their cell walls into gram-positive and gram-negative bacteria. Gram staining is the first step in the preliminary identification of a bacterial organism. Staining mechanism. Select a clean slide free of grease and transfer cells from a fresh culture onto the slide. The cells should form a thin, barely visible film. It is important to use fresh cultures as cells age, they lose their ability to retain the stain. Allow the slide to dry. Fix the cells to the slide by passing it over a flame of a Bunsen burner for 3 to 4 times. This may kill some bacteria but will fix the cells to the slide. After passing above the flame, the slide should feel warm when touched with the back of the hand but not too hot. Stain the fixed cells with a primary dye, crystal violet, and allow it to react for 30 to 40 seconds. Rinse the slide with water to remove excess stain. At this point, all cells appear purple under the microscope. Next, add a solution of Lugol's iodine and allow it to rest for 1 minute. The iodine combines with crystal violet to form diiodine complex to decrease solubility within the cell. Hold the slide at an angle and add ethanol or acetone dropwise until the drop coming from the edge of the slide just starts to become colorless. Wash off the excess ethanol. This is the differential step. Gram-positive bacteria retain the crystal violet dye, whereas gram-negative bacteria do not. When viewed under the microscope, gram-positive cells appear purple and gram-negative cells are colorless. Color the slide with safranin and allow it to react for 20 to 30 seconds. This stains the gram-negative bacteria pink. Rinse with water and dry the slide using a filter paper. When viewed microscopically, the gram-positive bacteria are purple and the gram-negative bacteria are pink. Principle: the difference in gram staining occurs due to the differences of cell wall composition of gram-positive and gram-negative bacteria. A gram-positive cell wall contains a thick layer of peptidoglycan with numerous teichoic acid cross-linking which resists the decolorization. In aqueous solutions, crystal violet dissociates into Cv+ and Cl- ions that penetrate the wall and membrane of both gram-positive and gram-negative cells. The Cv+ ions interact with negatively charged components of bacterial cells, staining the cells purple. The iodine, when added, interacts with Cv+ to form crystal violet iodine complexes within the cytoplasm and outer layers of the cell. The decolorizing agent like ethanol or acetone solution interacts with the lipids of the membranes of both gram-positive and gram-negative bacteria. The outer membrane of the gram-negative, that is, the lipopolysaccharide layer of the cell is lost, leaving the peptidoglycan layer exposed. gram-negative cells have thin layers of peptidoglycan. With treatment of ethanol, the walls become leaky and allow the large CVI complexes to be washed from the cell. The highly cross-linked and multi-layered peptidoglycan of the gram-positive cell is dehydrated by the addition of ethanol. The multi-layered nature of the peptidoglycan, along with the dehydration from the ethanol treatment, traps the large CVI complexes within the cell. After decolorization, the gram-positive cell remains purple in color, whereas the gram-negative cell loses the purple color and is only revealed when a counterstain, the positively charged dye safranin, is added.